Hello guys, this is the second video on the cell part of the biology in preparation for the IMAT. Today I just did my registration, and I believe by today you should all have finished your registrations. Good luck, nice. I hope you study hard, and I hope we all get into the universities we want. Now, I'm sorry for the delay, and I'm sorry for not uploading the videos. I was on vacation, so that's all. Now let's start right away. I want to remind you from the previous time what is compartmentalization. Now you see, organelles themselves are often surrounded by membranes. Last time we looked at the membranes function and the membranes function, structure, and all of that. Okay, most organelles have the same function, the same structure of the membranes, but on the organelles inside the cell, the membranes are a little bit thinner than the membrane, than the plasma membrane, the cell surface membrane. Okay, now the organelles are all surrounded by membranes. Okay, so that their activities can be separated from the surrounding cytoplasm. This is described as compartmentalization, like little apartments in a building block, something like that. This is what compartmentalization means. Now, you see, most organelles are compartmentalization and division of labor within the cell are even more obvious with an electron microscope than with a light microscope. In this PowerPoint, I believe I do not have any electron photos, because I don't think we should go much that in depth. I only have the pictures, the diagrams of the organelles. Okay. Now, we will now consider the structure and function of some of the cell components in more detail. First of all, let's start with a nucleus. This is not a cell, this is the nucleus of a cell. Okay. First of all, the nucleus is the largest cell organelle. It is surrounded by two membranes known as the nuclear envelope. You see, this is one membrane here and one here. These two, they join in some parts and they create these holes that are called nuclear pores. Of course, these holes are not open. Okay, yes, I will talk later about the nuclear envelope. Okay, now, the outer member of the nuclear envelope is continuous with the endoplasmic reticulum. We will see that later. Now, within the nucleus, the chromosomes are in a loosely coiled state, known as chromatin. In the next videos, we will talk about the cell cycle and how the genetic material is in all the phases of the cell cycle. You will understand what is chromatin and what are chromosomes and what is the difference between them. And now I want you to know that it contains the DNA. Okay, nucleus contains the DNA, contains the genetic material of the body, of the cell. Okay, now the DNA, chromosomes contain DNA, which is organized into functional units. They're called genes. We'll see later that genes control the activities of the cell and inheritance. Thus, the nucleus controls the cell's activities. When a cell is about to divide, the nucleus divides first, so that each new cell will have its own nucleus. Okay? Now, the nuclear envelope, we said that the outer membrane is continued with endoplasmic. There are some pores, some holes that you see how they join the two membranes. They join in order to create these holes. But these are not open holes. There are proteins in those holes that are blocking the holes and control which substance can go in and out, and they allow and they don't allow many substances to pass through, very specific ones. Okay, and examples are mRNA and ribosomes. Now you will ask, why ribosomes go in? You see, ribosomes are made of two units, a small subunit and a larger subunit, and rRNA. This rRNA is transcripted. We will see later what is that. Is created, let's say, inside the nucleus. Therefore, the small subunit goes into the nucleus to connect with this rRNA, okay, to create the complete small subunit. Therefore, ribosomes are created inside the nucleolus. What is that? You see, it is this black area. This is an electron photograph. This is the nucleus, you see. This is the nucleolus. Here is RNA is synthesized. The small and large subunits are assembled here, and it's the part of the DNA that is more cold, more it is more compact. The nucleolus is more darkly stained, as you see, because it is more dense. Okay, and the chromatin is more coiled in this area of the nucleolus. We will look at that later. Okay, it is not a membrane-bound structure, it is just an area of the nucleus where DNA is more coiled, is more tight. You can see in this picture that the outer membrane continues with the endoplasmic reticulum. And now we'll talk about ribosomes. You see, ribosomes are the site of protein synthesis. They are 25 nanometers in diameter, and they're made up of RNA and rRNA, which is R for, let me show you that, pen. R is from the word ribosomes. So R ribosomal RNA and proteins. So if we combine rRNA plus proteins, we get these two units which together create a ribosome. You see, we weigh them in how much time they take in order to sit in a, oh my god, I forget the word right now. Okay, I will come back to this later. Anyway, it's not important what I was going to say. Let's continue. They are found free in the cytoplasm and on the rough endoplasmic reticulum. ER is endoplasmic reticulum. Okay, I want you to know that they are not all the time together, and at a point the large subunit comes and joins with the small subunit. Actually, the process is the small subunit is here, the mRNA, which is about to be translated into proteins, comes in, and then the large subunit comes and hugs, let's say, the small subunit with mRNA. Okay, now let's continue. The endoplasmic reticulum, or as it is called, ER, are membranes, is a system of membranes. Okay, and there are two types. There is rough ER, endoplasmic reticulum, rough endoplasmic reticulum, and smooth ER. You see, rough ER has holes where ribosomes exist in those holes. Okay, so these holes, they make their texture more rough, therefore smooth, which does not have ribosomes attached to it. It is called smooth. Okay, they have completely different function. They may have the same name, but they don't have the same function at all. The smooth ER, which is the easiest one, it makes lipids and steroids. Okay, cytoskeleton, the reproductive hormones, estrogen and testosterone. Also, it stores calcium ions in muscle cells to facilitate normal muscle contraction. Actually, in the muscle cells, it has a specific name. And also, it detoxifies drugs and poisons from the body, like alcohol. Okay. Now, the RAF-ER. You see, we said that ribosomes is the site of protein synthesis, that they're found free in the cytoplasm and on the ER. But what is their difference? Do I have a full cell picture here? I don't. Oh god, wait, give me a minute, guys. I'm sorry. I want a full cell picture to show you something. Yes, let's see here. There are ribosomes here, okay, and on the RAF-ER. If a protein is synthesized on this ribosome, which is free in the cytoplasm, it will stay inside the cell. But if a protein is synthesized in the RAF-ER, in the ribosomes that are attached to the RAF-ER, then it will be secreted out of the cell. And this is the major difference between the two types of ribosomes. They decide, according to where they are located, where the protein is going to be used inside the cell. I'm sorry, inside the cell or outside of the cell. For instance, there are cells that their role, their major function is to secrete special proteins. Therefore, they will have a well-developed RAF endoplasmic reticulum and few ribosomes, because they do have needs and they do need proteins for themselves as well. Okay, so the RAF endoplasmic reticulum, is where proteins are synthesized to be exported out of the cell. Also, inside the RAF-ER, they are often modified. Proteins are often modified in some way. Okay. Now, there is the Golgi apparatus, or Golgi body, or Golgi complex. It's a stack of flattened sacs, as you see here. A cell may have one or more than that. Okay, the stack is constantly being formed at one end from vesicles, with bed off from ER and broken down again at the other end to form Golgi vesicles. You will understand what I'm saying later. Now, the Golgi body collects, processes, and sorts molecules ready for transport in Golgi vesicles, either to other parts of the cell or out of the cell. Secretion. Two examples of protein processing are the addition of sugars to protein to make molecules known as glycoproteins, and the removal of the first amino acid, methionine, from newly formed proteins to make a functioning protein. In plants, enzymes in the Golgi body convert sugars into cell wall components. Golgi vesicles are also used to make lysosomes, which we'll see now. The secretory pathway. Let me explain that to you. Here. Not here. Yes, here. You see, these are what? Proteins, okay? These dots are ribosomes. Let's say protein is synthesized here. So it's about to move here, and we will create in this vesicle, that this vesicle will be disconnected, will bud off from the ER. And this will be connected then here to this one. Okay, then it will move through this, and then it will go out. So we have a protein, it will get in here, it will move through the Golgi body to be modified, and then it will what? It will bud off. Therefore, you see this membrane, actually the Golgi body has two phases, the trans phase and the cis phase. And incoming transport vesicles come here, it connects with that, then it moves through that, it gets out from the trans phase in a secretory vesicle. And we've got this process that it goes out like this, the protein. Now this protein may do two things. If this secretory vesicle is maybe exported out of the cell by exocytosis, we have mentioned this earlier, but I have not explained you what it is. It's that. You see, this is exocytosis. It's released out of the cell, so this is a secretory vesicle. But if this secretory vesicle stays in here and joins with enzymes, we will have what is called a lysosome. What are lysosomes? Are spherical sacs surrounded by a single membrane and having no internal structure. They are commonly 0.1 to 0.5 micrometers in diameter. They contain digestive enzymes which must be kept separate from the rest of the cell to prevent damage from being done. Okay, they are responsible for the breakdown, which is the digestion of unwanted structures such as old organelles or even whole cells, as in mammary glands after lactation, breastfeeding. Okay, also in white blood cells, lysosomes are used to digest bacteria. Enzymes are sometimes released outside the cell, for example, in the replacement of cartilage with bone during development. The heads of sperm, they contain a special lysosome, the acrosome, for digesting a path to the ovum. Okay, and how are they created? Here it is. You see, let's say a food comes in by phagocytosis. We've said that the food vacuole will connect with what? Special enzymes from the Golgi body. They have been created where? In the rough ER. So we've got enzymes which are created by the ribosomes of the rough ER. They go through the Golgi apparatus and they're not lysosomes yet. When they connect with that, they are lysosomes. These lysosomes will destroy this food, or they will break it down, and therefore will have done their purpose, or a cell, a bacteria may come in, and this process will happen in the white blood cells. Okay, this is the anatomy of lysosome. You see they have hydrolytic enzymes. That's all. Now, mitochondria. The structure. Mitochondria are usually about 1 micrometer in diameter and they have various shapes, but they are often sausage shaped, like that. They are surrounded by two membranes, an envelope. But you see, where am I? Yes, they're surrounded by two membranes. The number of mitochondria in a cell is very variable. As they are responsible for aerobic respiration, it is not surprising that cells with a high demand of energy, like liver cells and muscle cells, contain large numbers of mitochondria. When you say large, about what? The 5 mitochondria a cell? 20 mitochondria a cell? How many? As many as 2,000 mitochondria. So if you exercise regularly, your muscles will make more mitochondria because they have more energy needs. Now, the inner, we said that mitochondria have two membranes which form an envelope. The inner of this is folded to form a finger-like criste. This is a criste. and this is the inner membrane. You see how it is being folded? Why is it folded? We will see why. The space between the two membranes here is called intramembrane space. The inner membrane is a far more selective barrier and controls precisely what ions and molecules can enter the matrix. The matrix is the interior solution. Here, this is the interior solution, the matrix. Now you see the main function of mitochondria is to carry out aerobic respiration, although they do have other functions such as synthesis of lipids. What is aerobic respiration? During respiration, a series of reactions takes place in which energy is released from energy-rich molecules such as sugars and fats. Most of this energy is transferred to molecules of ATP. ATP is adenosine triphosphate, and it's a universal. It is known as the universal energy carrier, and is the energy-carrying molecule found in all living cells. The reactions of respiration take place in the solution in the matrix and in the inner membrane, the crista. The matrix contains enzymes in solution, including those of Krebs cycle, and these supply the hydrogen and electrons to the reactions that take place in the crista. The flow of electrons along the precisely placed electron carrier in the membrane of the crista is what provides the power to generate ATP molecules. Now, why does the crista fold? The folding of the crista increases the efficiency of respiration because it increases the surface area available for these reactions to take place. Now, this is what happens. Once made ATP leaves the mitochondria, and as it is a small soluble molecule, it can spread rapidly to all parts of the cell where energy is needed. Its energy is released by breaking the molecule down to ADP plus phosphate. ADP is adenosine diphosphate, which is triphosphate. Therefore, one phosphate group leaves, and here it is. So this breakdown, which needs water, therefore it is a hydrolysis reaction, releases energy. The ADP now goes back to the mitochondrion, where it connects with phosphate ions, and it converts back to ATP during aerobic respiration. Now let's see what is the endosymbiont theory. You see, in the 1960s, it was discovered that mitochondria and chloroplasts contain ribosomes, which are slightly smaller than those in the cytoplasm and are the same size as those found in bacteria. The same of ribosomes, what I wanted to say earlier, is that it is measured in S units, which are a measure of how fast they sediment in a centrifuge. Cytoplasmic ribosomes are 80S. You see, 80S. While those of bacteria and mitochondria are 70S. It was also discovered in the 1960s that mitochondria and chloroplasts contain small circular DNA molecules, also like those found in bacteria. It was later proved that mitochondria and chloroplasts are in effect ancient bacteria, which now live inside larger cells, typical of animals and plants. This is known as the endosymbiont theory. Endo means inside, and symbiont is an organism which lives in a mutually beneficial relationship with another organism. The DNA and ribosomes of mitochondria and chloroplasts are still active and responsible for the coding and synthesis of certain vital proteins. But mitochondria and chloroplasts can no longer live independently because most of the proteins needed for their function are being coded by the nuclear DNA. They have DNA. Now let's see what are peroxisomes. Peroxisomes, they contain an enzyme which is called catalase that converts hydrogen peroxide, which is toxic, okay, the waste product of respiration in the cell, into water with the release of oxygen atoms. They also detoxify alcohol in liver cells. And so we start. So there is a joke in order to remember that H2O2 is poisonous or toxic. It says the bartender asks two scientists walk into a bar. The bartender asks what they would like to drink. The first scientist says, "I'll have a glass of H2O, please." And the second scientist says, "I have a glass of H2O2." The bartender understands this, H2O2. Therefore, the second scientist dies. This is supposed to be a joke. I don't find this funny, but it is good in order to remember that H2O2 hydrogen peroxide is poisonous. It is toxic. Okay? Let's move on now. Microvilli, singular microvillus. What are they? They are finger-like extensions of the cell surface membrane, typical of certain epithelial cells. Epithelial cells are cells that cover surfaces of structures. They greatly increase the surface area of the cell surface membrane, and this is useful, for example, for absorption in the gut or for reabsorption in the proximal convoluted tubules of the kidney, something we will look into later. Now, these are cells. You see, this is the nucleus here. Nucleus, nucleus, nucleus. This is in the small intestine. This is the lamen. The lamen is the area inside the small intestine. You see, there are microvilli. Its cell has what? These extensions, which are called microvilli, like hair. Imagine it like long hair, and they are useful for those cells, these epithelial cells. Okay. Now we're going to talk about the cytoskeleton. Give me a second. So the cytoskeleton is a complex mesh of microfilaments that extends throughout the cytoplasm. It has several important roles. First of all, it maintains the cell's shape. Second of all, it controls the position of organelles within the cell by anchoring them to the plasma membrane. There is an M that is missing here, anchoring them, okay, to the plasma membrane. It is involved in the flow of the cytoplasm, known as cytoplasmic streaming. And it anchors the cell in place by interacting with extracellular components. Now, the cytoskeleton, it includes microtubules and microfilaments. Now, let's see what, you see, this is the cytoskeleton. You see all those messy parts? This is the cytoskeleton. This is a microtubule, and this is a microfilament. Okay, what are microtubules? They are long, rigid, hollow tubes. They are very small, about 25 nanometers in diameter. Together with actin filaments and intermediate filaments, they make up the cytoskeleton. Okay, and essential structural component of cells. Now, microtubules are made of a protein called tubulin. This protein has two forms, alpha tubulin and beta tubulin. Alpha and beta tubulin molecules combine to form dimers. This is the alpha tubulin, this is the beta tubulin. You see, they form this dimer. These dimers are then joined end to end to form a long protofilaments. This is an example of polymerization. 13 protofilaments then line up alongside each other in a ring to form a cylinder with a hollow center. This cylinder is the microtubule. You see, we've got 13 protofilaments. Okay. Therefore, there is a helical partner. Now, apart from the mechanical function of support, microtubules have a number of other functions. Secretory vesicles and other organelles and cell components can be moved along the outside surfaces of the microtubules, forming an intracellular transport system. Membrane-bound organelles are held in place by the cytoskeleton. During nuclear division, the spindle used to separate chromosomes during mitosis and meiosis. Also, cilia and flagella, which move cells from one place to another, consist of 9 pairs of microtubules organized around 2 singlet microtubules. Okay, no. Spindle fibers help to separate chromosomes, we send that, and flagella, when present in prokaryotes, are not made of microtubules though. Now, the assembly of microtubules from tubulin molecules is controlled by special locations in cells called mTOCs, or microtubule organizing centers, therefore mTOCs. Now, we will discuss further. We will talk about centrioles, but because of their simple construction, they can be formed and broken down easily. Microtubules can be formed and broken down easily at the mTOCs, according to need. Now, microfilaments, what are those things? They are assembled from actin filaments and help support the shape of cell. This is microfilaments. Okay. Yeah. They enable animal cells to form a cleavage furrow during cell division. Amoeba to move by sending out pseudopods, and skeletal muscle to contract as they slide across myosin filament. We will look at the third one in more detail. That's all. Now, what is a centriole? The X-ray resolution of the electron microscope reveals that just outside the nucleus here, there are two centrioles. What is a centriole? They lie close together and at right angles. This is one centriole, another, and together they form the centrosome. Now, these are absent in most plant cells. Now, a centriole, what is? It is a hollow cylinder, about 500 nanometers long, formed from a ring of short microtubules. Okay. Each centriole contains 9 triplets of microtubules. The function of the centrioles remained a mystery. Until recently, it was believed that they acted as mTOCs. What is mTOC? That thing here. Microtubule organizing centers. Okay. For the assembly of the microtubules that make up the spindle during nuclear division. It is now known that this is done by the centrosome, but does not involve the centrioles. Okay. They are found at the bases of cilia and flagella, where they are known as basal bodies, and they do act as mTOCs there. The microtubules that extend from the basal bodies into the cilia and flagella are essential for the beating movements of these organelles. Now, the centrosome are two centrioles oriented at right angles to each other that make up one centrosome that consists of 9 triplets of microtubules. Each centriole consists of 9, 1, 2, 3, 4, 5, 6, 7, 8, 9 triplets of microtubules, and we have a centriole. Two centrioles at right angles, 90 degrees here, is a centrosome. Okay, plant cell. The centrosome acts as mTOC, but plant cell lacks centrosomes but do have mTOCs. Okay, in animal cells the mTOC is synonymous with centrosome. Now let's talk about chloroplasts. The structure of the chloroplast, as seen with the electron microscope, is visible in figures here. Okay. Now, the chloroplasts have an elongated shape and a diameter about 3 to 10 micrometer. Do you remember the diameter for mitochondria? It has only 1 micrometer, while the chloroplast 3 to 10. Like mitochondria, they are surrounded by two membranes, you see, inner and outer membrane, forming the chloroplast envelope. Also like mitochondria, chloroplasts replicate themselves independently of cell division by dividing into two. The main function is to carry out photosynthesis. Chloroplasts are an excellent example of how structure is related to function. So we'll look into their function in order to understand their structure. This is photosynthesis. I have this in words, the first and the second, and I will talk about it here. Now, during the first stage of photosynthesis, the light-dependent stage, light energy is absorbed by photosynthetic pigments. That means that light enters chloroplast, okay. But by photosynthetic pigments, particularly the green pigment chlorophyll. Some of this energy is used to manufacture ATP from ADP. An essential stage in this process is the splitting of water into hydrogen and oxygen. See, so we've got chlorophyll grana that captures the light energy. We'll see what is that. Chlorophyll exists in these things that are called grana. These together, we'll look at it, okay? And some of this energy is used for the splitting, okay, of water into hydrogen and oxygen. The hydrogen is used as fuel, which is oxidized to provide the energy to make the ATP. This process, as in mitochondria, requires electron transport in membranes. These explain why chloroplasts contain what? A complex system of membranes. The membrane system is highly organized. It consists of fluid-filled sacs called thylakoids. You see, this is thylakoids, which spread out like sheets in 3 dimensions. In places, the thylakoids form flat disc-like structures that stack up like piles of coins many layers deep, forming structures called grana. Grana means grains, which is their appearance in light microscope. These membranes contain the photosynthetic pigments and electron carriers needed for the light-dependent stage of photosynthesis. Both the membranes and whole chloroplast can change their orientation within the cell in order to receive the maximum amount of light. Now, in the second stage of photosynthesis, the light-independent stage here, this second stage uses the energy and reducing power generated during the first stage here to convert carbon dioxide that enters the cell into sugars. These require a cycle of enzyme-controlled reactions called the Calvin cycle and takes place in solution in the stroma. What is the stroma? Is this, like the matrix in mitochondria. Okay, the solution inside the inner membrane. The sugars made may be stored in the form of starch grains in the stroma. If you don't remember what starch is, go back to the biochemistry videos when I talk about carbohydrates. Okay, and actually, no, polysaccharide, when I talk about polysaccharides. Starch is a polysaccharide. Okay, the lipid droplets, also seen in the stroma as black spheres in the electron micrographs, are reserves of lipid for making membranes or from the breakdown of membranes in the chloroplasts. I don't believe I've got anything here. Now, as with mitochondria, it has been shown that chloroplasts originated as endosymbiotic bacteria, in this case photosynthetic blue-green bacteria. Okay, again, the endosymbiont theory applies to mitochondria and chloroplasts. Now we're going to look at two fundamentally different types of cells. At one time, it was common practice to try to classify all living organisms as either animals or plants. With advances in our knowledge of living things, it has become obvious that the living world is not that simple. Fungi and bacteria, for example, are very different from animals and plants and from each other. Eventually, it was discovered that there are two fundamentally different types of cells. The most obvious difference between these types of cells is that the one possesses a nucleus and the other does not. Does that mean that if it doesn't have a nucleus, it does not have DNA? Of course not. It has DNA in an area of the cell, okay, but it is not contained in a nucleus. There's no nuclear envelope. It is free in an area of this type of cell. These type of cells that lack nuclei are called prokaryotes. Pro means no, before, I'm sorry, and karyon means nucleus. They are on average about 1,000 to 10,000 times smaller in volume than cells with nuclei, and are much simpler in structure. For example, their DNA lies free in the cytoplasm. Organisms whose cells possess nuclei are called eukaryotes. Eu means true. Their DNA lies inside a nucleus. Eukaryotes include animals, plants, fungi, and a group containing most of the unicellular eukaryotes known as prokaryotes. I don't believe you should know all that. This is why I don't write all of that. Okay. Now, most biologists believe that eukaryotes evolved from prokaryotes 1,500 million years after prokaryotes first appeared on Earth. We mainly study animal and plants for IMAT, but all eukaryotic cells have certain features in common. Now, generalized, here's a difference between prokaryotes and eukaryotes, a table that I think you should know, but I won't go into all of them. I found the updated picture with much better quality, so I will upload this one. And last but not least, I'm going to talk about viruses. Okay. In 1852, a Russian scientist discovered that certain diseases could be transmitted by agents that, unlike bacteria, I forgot something. Bacteria are prokaryotic organisms. Therefore, you need to know some things about prokaryotic organisms because you need to know some things about bacteria. Okay? I'm sorry, this is my dog that makes this sound. Now, in 1852, a Russian scientist discovered that certain diseases could be transmitted by agents that, unlike bacteria, could pass through the finest filters. This was the first evidence for the existence of viruses, tiny organisms which are much smaller than bacteria and are on the boundary between what we think of as living and non-living. You see, unlike prokaryotes and eukaryotes, viruses do not have a cell structure. In other words, they are not surrounded by a partially permeable membrane. containing cytoplasm with ribosomes. They are much simpler in structure. Most consist only of a self-replicating molecule of DNA or RNA which acts as a genetic code and a protective code of protein molecules. We see the structure of a simple virus. Actually, this virus here that I show you is the virus that causes the AIDS, type of retrovirus that causes AIDS, HIV. Human immunodeficiency disease is HIV, and we will talk about that in a later video. Now, you see, it has a very symmetrical shape. Its protein code, or as it's called, capsid, is made up of separate protein molecules, each of which is called a capsomere. Okay. Viruses range in size from 20 to 300 nanometers, about 50 times smaller on average than bacteria. All viruses are parasitic because they can only reproduce by infecting and taking over living cells. The virus DNA or RNA takes over the protein synthesizing machinery of the host cell, which then helps to make new virus particles. And this is why they're parasitic. They use another living organism to live. In this video, I'm going to post a test in the comment section that you could take to refresh your knowledge. I want you to know I have posted another one in the biochemistry part 3, and the answers are also there. They have been updated. And I want you to know that sometimes in those tests there are questions that I haven't mentioned before. This is why, because after I make this video, you see, I found out something in a book and I said, oh God, I should have mentioned that, and why didn't I? And I can't make a video just to mention one sentence. So if I put a question in the test that you don't know, don't be like, oh my God, I wasn't correct on the videos. It's because I chose to put it there in order to learn this new thing. Okay, thank you so much, and hit the subscribe button if you want to be updated when I upload new videos. Bye bye.